Hello. So before we start, I have a quick announcement by a person who's involved with the labs, Lloyd Goldwasser. So there you go.
Thank you. Good morning. I'm part of a team that's revising the Biology 1B Evolution Labs. This is good news, and you can help. We want to know about your experience and your thoughts about the Evolution Labs, what you bring to them, and what you get from them. We're conducting an online survey once now before the labs start, and again later this semester, and we encourage you to take 15 minutes to fill it out. It's completely voluntary. You can stop at any time, but because you're helping future biology students, if you complete the survey, you'll receive a free extra credit bonus point towards your grade in the class. You'll get a point for completing the first survey now, and you'll get a point if you complete the second survey later this semester. To make it truly voluntary, we offer an alternative way for you to get the bonus point. It involves reading an article about science lab practices and writing a summary of it. You have to be at least 18 years old to do the survey. If you're less than 18 and want to get the point, then you have to do the alternative task. And your responses will be completely confidential. We ask for your student ID to be able to give you the bonus point, but then we remove your ID and replace it with the randomly generated ID. Once that's done, there is no way to connect any responses with any specific individual. If you've looked at your email in the last few hours, you may have seen that the email invitation has already been sent out. It contains a link that will take you to the survey itself, and it will be open until the evening of Sunday, September 9th. After that point, we can't use your responses, so you won't get the extra credit. So the time for you to do the survey is as soon as possible. If you don't receive an invitation, or if you have any questions, please email me or Tammy Mao. Here are our email addresses. Thank you very much. See you later.
Okay. Can you hear me again? Can the people in the back hear me? All right. I can't see you very well right now, so assuming you're saying yes. So thanks in advance if you go ahead and do this survey. It's very helpful to us. Just as an aside, this is I don't know what you thought of Roy's presentation. I thought it was fine in terms of obviously get some free points. That's probably what the thing you mostly heard is. But on the other hand, it's interesting because it turns out you guys are humans and you're human subjects as far as the government's concerned. So we have to be very careful about if we're doing a test on you, that's an experiment. And this is a sort of experiment because we want to see how people respond to the current sets of labs, the before and after questions. And then we'll be doing a similar type of survey when we roll out the new labs, the before and after responses. So this is sort of an example of a controlled experiment, in as much as at least we're asking a cohort of 850 students, they're find out what they've learned, what they came in with, and what they learned in the current labs, and what they come away with in the future new labs that will be rolled out next semester. So we appreciate your response. And if that sounds weird, because there's lots of protocols working with human subjects, as it turns out. So anyways, I thought you'd be interested in that. I want to continue today with population genetics. I decided to put on the board just a little bit of a flow diagram, or like a simplified history of evolutionary biology from the publication on the origin of species in 1859 by Darwin, and what happened in the preceding years. I told you in the last lecture that initially some aspects of this theory were accepted immediately by most people, at least most practicing scientists, such as the idea that evolution occurred. Yet amassed so much additional evidence that this old idea people were convinced by it. But many biologists argued about whether natural selection was by itself sufficient to cause the patterns we see in the rock record or out there in the world. So they wondered or argued whether or not natural selection by itself was a sufficient process. So there were these series of arguments. There were also arguments, as I mentioned, about how does inheritance work, right? And that was sort of it was a key part of the theory, in as much as you had to have inheritance in order to have natural selection. But in terms of the mechanism, at least Darwin had no idea. It turns out, like I mentioned, Mendel had something to say about how inheritance works. He argued that it was particulate. That is to say, there are bits that you inherit from mom and dad that code for traits. We'll talk more about that in a bit. Also, after Mendel, but in the early 1900s, the field of population genetics started up. Okay, this was founded by a number of very smart people whom I'll discuss in a bit. But the field of this mathematical theory that describes how alleles change their frequencies in populations and various evolutionary forces was developed in the early 1920s. And it's still a very vigorous and exciting field today, at least in my mind. And the reason why population genetics even came into being is because of a very simple reason. And that reason is as follows: humans, we live a pretty long time compared to other organisms, but we still only live, let's say, 80 years. And a research career such as mine might span, well, I'd like it to span as long as possible, but let's say 40 years, right? And a particular project that might be supported by the government, a typical government grant, for instance, lasts 4 years. So how much evolutionary change can you observe over the course of, say, 40 years or 4 years, right? You can observe some, and people have. There's lots of very neat experiments that have been done out in places like the Galapagos Islands and other places, especially in rapidly evolving organisms, where you see change over the course of an NSF or NIH grant. But the general problem is that even when natural selection, when it acts weakly, can be a very powerful force on a geological timescale, even though it would be difficult to observe over the course of a human lifetime. So this mathematical theory was developed largely to address questions like, if an allele confers a very small selective advantage, what type of changes would we expect to see over time? We expect to see the allele change its frequency by a large amount over 10 generations, 1,000 generations, 1 million generations. What is the time scale of the change? So we can actually extrapolate into the future what types of allele changes in allele frequencies we might expect with different evolutionary forces working. That was why mathematical theory was developed. Around in the 1930s and 1940s, the so-called modern synthesis, or the neo-Darwinian synthesis occurred. It basically took several different branches of knowledge and incorporated these different branches of knowledge into modern evolutionary biology. So, for instance, this is where genetics was formally incorporated into phylogenetics. This is where biogeography and the observations from relationships amongst different species was incorporated into evolutionary biology. And since then, there have been many different avenues of research, and it's a very active field, as I mentioned earlier. For instance, Watson and Crick, you're probably aware that they discovered the structure of DNA in the 1953, I believe, is when they announced it. And that there's a very exciting field. I'm involved in this field of molecular evolution, which I think is exciting. Molecular evolution, how can we explain patterns, differences among genomes in different organisms? What types of evolutionary forces shape genomes? There have been fields such as experimental evolution that have thrived, comparative studies where you compare one species to another and look at the characteristics to infer what types of evolutionary forces have occurred in those species in the past. That's a very, very exciting and vigorous field. But we're going back to Mendel, so I just want to give you that overview. Mendel left with the idea that Mendel had these ideas of locus, which you can think of as a gene, if you will, and we had this idea of alleles. These are variants at a locus, different variants of a locus. And a locus could be almost anything that you define it as. So if you're a geneticist, you can define a locus however you want. I mentioned in the last lecture that one type of locus that you'll hear perhaps about in newspaper articles they're called SNPs. SNPs. What is a SNP? It's a single nucleotide polymorphism. The idea is that the resolution that we have now with modern genome sequencing techniques allows us to find single nucleotide positions in a genome that differ from one individual to the next. Right? And these SNPs are becoming very important, or experiments or surveys using SNPs are becoming very important in discovering the causes of many diseases today. Okay? But SNPs would be examples of alleles. The locus might be a particular position in the genome, so you can number those however you want, but usually it's which chromosome it's on, and you can label the number of the nucleotides on a chromosome from one end to the other. So it'll be a particular nucleotide position, and then the variance of that nucleotide position, well, there's only 4 possible variants, right? You can either have A, C, G, or T at that position. So some individuals in this room might have one variant for that for a particular SNP, maybe have an A at a particular position, and other individuals in the room might have a different nucleotide at that same position, a G. That would be examples of an allele. In this case, it would be a SNP allele, SNP allele, single nucleotide polymorphism. And that would be, you might imagine that would be the most resolution you can expect in any type of genetic experiment, right? So you're not going to find anything more basic than a SNP. Okay? But anyway, so I just want to give you some ideas of, you probably already heard of locus and alleles from your high school biology, and just give you some ideas of what these might be in modern terms. Now, in population genetics, it's a very mathematical field, and if you're going to do math on something as messy as biology, you have to understand an important distinction that modelers have to make. Modelers being mathematicians that try to model complex processes. And the idea is that you're going to use models. Models are idealizations of what's really out there. And there's one of my favorite quotes from a scientist, actually, from a statistician by the name of George Fox, is as follows: all models are wrong, but some are useful. Right? So a model doesn't have to be a perfect description of nature to be a good model. In fact, a model that's a perfect description of nature is as complicated as nature, and it's not a simplification, right? And it becomes useless. Okay, so as an example, a good example of the model, if you've ridden the BART, right, and you look on you go to the train station, sometimes they have these maps inside the cars. Of course, in the stations, the BART stations, they also have maps that show the different lines, the different BART lines, the different colors with their stops. Right? So you'll have something like this. You might have the Pittsburgh out here, Pittsburgh Bay Point, and then it goes there's one line, the yellow line that goes all the way to SFO, right? And then they'll have the stops marked, but with ticks, right? That is a model of the BART on one line of the BART. And of course, there's places where you have stations that you can actually cross the lines and so forth. This is a model, and it's an incredibly useful model if you want to get from, say, Berkeley, tells you to change at MacArthur, to the yellow line to get to SFO, right? So that is an incredibly useful model if you're going home for Christmas or something, and you're trying to get to SFO from Berkeley, through the Berkeley station, right? Another model would be, that would be one example. An alternative model would be a satellite photo of the Bay Area. Right? Which of those two models is more detailed and in some sense better represents reality? Well, I would say the photo from space of the Bay Area is a very good model in terms of its detail, the resolution. It has a lot of features on it, such as buildings and rivers that are very accurately mapped. But it's almost totally useless if you want to get from Berkeley to SFO on the BART, right? And the reason is it has so much extraneous information, it doesn't matter. You don't really care if the lines I drew here are perfect representation of the way the tracks go. It doesn't matter if these tick marks perfectly represent the distance from one station to another. They can be evenly spaced for all you care. You just want to know which is the next station. You don't want to know the distance, right? It's irrelevant for your trip. This is a good example of a very useful model, although it lacks a lot of details, right? Versus a model such as a satellite imagery, which is it could be a useful model for some cases, but it's certainly not a useful model if you want to get to SFO on BART. Right? I think it's probably pretty clear. So population genetics has this notion of a population, and this notion depends critically on idealization of what's out in nature. So I like to draw populations. I don't know why, but it's very convenient as circles, right? I sometimes think of them as almost petri dishes, but it doesn't matter. And in these populations, we have individuals. So here's an example of a population with individuals. Now, if you're a mathematician, one thing you might say is, well, what are the number of individuals in the population? Something that population geneticists always call n. Okay? That's the first notion. Here's an idealization of a population, and you already can tell it's an idealization because the geographic bounds of any population in nature typically aren't circles, and individuals aren't little circles, right? They're individuals, they're complex organisms. And really, from our perspective, we think of each one of these individuals as having alleles, maybe big A, big A, big A, little a, little a, little a, some locus. So we just think of the individuals as being carrying different alleles, potentially different alleles at some locus. And so it's a very, very simplified situation. And there's this other notion, another idealized notion that population geneticists use, called random mating. So I don't know what you're thinking about. You're probably thinking that population geneticists are somehow maybe hedonistic or whatever, but it's not that. The idea is that every individual within a population has an equal probability of mating with any other individual in the population. So if you want an example, would be as if some female in this class gave out to all the males in this class a ticket with a different number on it, and the person she ended up marrying, say, would be just draw the ticket from a hat. That would be random mating, right? It would be a very weird situation, but that would be an example. So clearly that's an idealization of what happens out in nature, right? Clearly men aren't assigned lottery tickets. But it's an example of how population geneticists think about populations. Now, this is how people think about populations in theory, as a model. And these models allow us then to do calculations on these populations. But then the next question is, well, say you're a practicing biologist, and you have this model. How do you apply this population genetic theory in practice? Because in real life, you go out, maybe you're interested in, I don't know what you're interested in, maybe you're interested in some species of bird, or maybe you're interested in gophers. I don't know, but let's say you're interested in gophers. So you go out, you sample gophers from the wild, maybe you go to the Sierra Nevadas, and you collect humanely as possible a bunch of gophers, and you type them at some locus. So now you have, for a bunch of different gophers, you know which alleles are present at one or more loci. What do you do? How do you define a population in nature? How do you determine whether you have the situation where you have a bunch of individuals that are randomly mating? Now, it turns out that how you go about defining a population in practice doesn't matter too much. It can matter some, but not too much. So, for instance, it is possible for a biologist or maybe an epidemiologist would be a better example to talk about the population of San Francisco. Right? Now, we all know that individuals within San Francisco aren't randomly mating with one from the other, and we also know that individuals from San Francisco aren't isolated in any way from other such populations. It's not as if people from San Francisco don't go across the Bay Bridge or the Golden Gate Bridge or highways 101 or 280, right? They can easily go from San Francisco to elsewhere. But there might be many problems you might be interested in, for which defining a population as those individuals who live in San Francisco, the city limits of San Francisco, might be a perfectly valid, reasonable way of defining a population. Maybe you're interested in the frequency of some allele, or maybe you're interested in the frequency of some disease in that region, right? And in practice, it seems to not matter too much how practicing biologists define the populations as well. One other thing I want to say about populations is that every species has at least one population. Right? There's always at least one population, idealized population in each species. That's not to say that you can't have more than one population in a species. So here's an example of a second population with another, maybe we call this N2, we'll call this the population size over here N1. And you can have movement of individuals from one population to the next. Individuals can migrate from one population to the next. Once again, this is an idealization that population geneticists think about, but it's a useful one, and it's one we'll be referring back to later in the class. Right? So every species has at least one population. Okay? And just understand the difference, or I hope you appreciate that you can have a model that is wrong in many of the details, that is to say, it doesn't incorporate many of the details that are out there, but is useful and correct in important respects, for the characteristics of the phenomena that you actually care about. Okay. So the first result I want to talk about in population genetics is a fairly old one. It's the first one, in fact. The first result I'm going to discuss is also the first population genetics result in the literature, about 1910. And it was independently developed by two different people. One was a German physician with the last name of Weinberg. And the other person who contributed to this theory was an English number theorist. If you know what number theory is, it's these people that try to prove results regarding things like prime numbers. Right? Hardy was one of the best number theorists ever. And so this theory I'm going to describe goes under the name of the Weinberg-Hardy theory. No, that's not right. The Hardy-Weinberg theory, right? Hardy-Weinberg. And it describes how alleles change frequency in a population under only the influence of random mating. Okay? Once again, you think about one population and you think about randomly mating individuals. Another characteristic, other assumption we're going to be making is that population sizes are large. And when I mean large, I actually mean infinity. Right? So once again, if you have math, once you have things formalized as equations, you can always think of n as going off to infinity. That's not important. The reason why we think about why it's important to say this is we're not accounting for random changes in allele frequencies with this theory. There's no random changes. So what does the Hardy-Weinberg theory do? It describes how alleles, what the genotype frequencies are in a particular generation under only the influence of random mating. And so I'm going to give a very simple example. I believe it can even be found in your book, at least it can be found in the 8th edition of your book, on page 473 in the 8th edition. I'll find out where it is on the 9th. But this example is just going to be with flower colors. And the flower colors are going to be the phenotype characteristic that can be directly observed in this case. And there's three different flower colors we're going to consider. They're going to be red, pink, and white. Okay. So I'm going to make a table out of this. Okay, so those are the three phenotypes. And each of these phenotypes has a genotype that's associated with it. Or maybe it's better to say each of these genotypes has a phenotype associated with it. So the red phenotype is caused by having been homozygous for a particular allele. So the allele is going to be called a C with a superscript R on it. Right? Now, you might ask, well, why not A, big A, for instance, or big B, or whatever you want. And the reason why it's C with a superscript R is because the person who first discovered this allele, they called it C superscript R. So if you're the person who describes an allele, you can call it whatever you want. Right? So CR, CR. So this is an example of allele 2. And this is the genotype. They're homozygous. If you have two of the same type of allele, then you're homozygous for that allele. The white phenotype is caused by having CW, CW. That would be homozygous for the white allele. Once again, it's named that because the person who discovered the allele named it that. And the pink allele is caused by being heterozygous for the CR and CW alleles. Those are the three genotypes. Now, in a population, if you were to go out into nature, you might actually try to survey the frequencies or numbers of each genotype. So we'll call them the genotype count. The idea being that you somehow define your population, maybe it's all the flowers in some meadow, right, as an example. And you went out and you had a way of assessing the genotype for each of these different, each flower you see. Now in this case, this example is a little bit unusual in at least one respect, and that is that if I tell you the phenotype of the flower, you can tell me the genotype. Right? It's not always that simple. Sometimes as an example, eye color, actually eye color is a more complicated trait than people usually think, but as an example, I'm just going to write, I'll do it over here. Brown eyes can be caused by being homozygous for the brown allele, or brown eyes can be caused by being heterozygous for the brown allele. And blue eyes, you have to be homozygous for the recessive allele. So in this case, just knowing the eye color is not sufficient to tell the genotype, right? But in this case it is. So you have some way, some fancy way perhaps, of determining the genotype at the locus. And so we have a count. We have 320 individuals that are CR/CR. We have 160 who are heterozygous, and we have 20 that are homozygous for the CW allele. And you note that the total number of individuals we sampled was 500, right? We have 500 individuals that were sampled. So I'm just going to continue this table on the other board here. We can have a genotype frequency. So this is just turning those counts that you directly observed in the population into frequencies. I told you we have 500 individuals, so the frequency of the CR/CR genotype is 320 divided by 500. And that should be 0.64. And the frequency of the heterozygous genotype is 160 divided by 500. And that equals 0.32. And finally, we have the frequency of the homozygous, the last genotype, the CW genotype, which is 20. The number of individuals by the total. And that should be 0.04. Okay. So that's just turning counts into frequencies, and I imagine that you guys, considering you're here at Berkeley, that this was something that you're familiar with. But if you're not, then you can certainly feel free to come to me. And just one quick check, even if you understand the principle, everybody realizes you can make mistakes mathematically. I think I've done this, made such mistakes in this lecture in the past. One quick check is to make certain these three frequencies sum to 1. They have to sum to 1. If they don't, then you may have made mistakes somewhere. All right, so far we're just doing things that you can just manipulate these counts. Another type, probably one of the most important things you need to be able to calculate are the allele frequencies. All right? The allele frequencies are going to be calculated from the observed genotype frequencies or counts. There's really